So today I'd like to tell you this is sort of a tutorial. Actually, I gave it as more of a software tutorial right there close to where you guys are at CGSI, which is in the engineering building this summer. And then I kind of adapted it to be more conceptual, more about open problems for this audience. But nevertheless, it's sort of based on some problems that we've been tackling. I'll present some of our solutions to it, and hopefully others will be inspired to develop more solutions. So, what motivated us to get started on this thread of development was or at least one of the motivations was that diseases involve these dynamic cellular state changes. What that means is technically you could have the same cell type. So in Alzheimer's, for example, you have these immune cells in your brain called microglia, and the cell type is the same, but they change from homeostatic to kind of a degenerative inflammatory state. Today I'll have a running example about breast cancer, which we're working on. The cells can start, even if they're cancerous, they start in the mammary tissue. Initially, they can be quite a benign cancer, and then they can transition to being sort of a metastatic or more invasive state. So these kinds of cellular state changes accompany almost every disease. And there could be some genetic background for it, but these states are largely defined based on genomics, epigenomics, and signaling. So there are some predispositions potentially due to mutations, but one of the things about these cell states is that they can also switch back and forth. So this is again kind of an example from triple negative breast cancer abstracted out. Initially, when the cancer starts, the cells are in more of an epithelial state, which your breast tissue is mostly epithelial. They can undergo an E to M transition to kind of a hybrid, EM hybrid type of state. And this is the state that most likely causes, it's thought, the metastatic invasion and secondary tumor formation. Whereas if they completely transition to mesenchymal, then we're a little bit safe yet. And there's things that can switch back and forth between these states. And in specific, therapies are thought to switch between these states. So sometimes the very therapies that you think are helping, like chemotherapy. Chemotherapy does cause some amount of cells to transition to a chemorefractory or resistant state that can be more pernicious. But hopefully in the future we can leverage these dynamic state changes. It also turns out, and we saw this in a different study on colorectal cancer, that the cellular microenvironment can also drive these state changes. So it turned out that in our colorectal cancer study that I'll reference later, we saw that fibroblasts were having sort of a protective effect on cancer cells and not allowing therapies to be effective on them. Cancer metastasis, which I mentioned before, often involves these transitions. And I think you already heard from other speakers that metastasis is probably the most fatal part of cancer. If it was just the primary tumor, then you would just excise it and be done. But in metastasis, it could spread to multiple organs, and then it kind of gets out of control. And, you know, the way these states are characterized is by what genes the cells are expressing. Currently, people know, for example, in triple negative breast cancer, again, that epithelial markers, ecdierin, laminin, and things like that, beta-catenin, are expressed in the more epithelial-like state. And then when it changes to the more mesenchymal-like state, you get completely different markers, different behavior too. The epithelial states are adhered to each other. I think the cadherin helps it. And then they kind of escape from one another, change shape, become modal. A lot of changes are happening. And these aren't necessarily genetic, so they don't have to be accompanied by accumulation of mutations. And recently, this has been known under this term that some people find nebulous called cancer plasticity. In some of the studies that we've read, plasticity refers to the ability to switch states. It's sort of like the potential for the cell to switch states. So sometimes it's thought that chemotherapy increases the plasticity. It allows new states to be reached from the normal state. And then the idea is, could we have certain kinds of therapies that are currently speculatively referred to as state gating therapies that might curtail this plasticity and keep cells in certain kinds of states, gate out certain other kinds of states? So cellular plasticity is now widely recognized as a so-called hallmark of cancer. This is from the Hanahan updated cancer hallmarks. And the hallmarks of these cancers, there's like many of them. And they're often future targets, so things like non-mutational epigenetic reprogramming. But plasticity also is recognized as a hallmark of cancer, and there's not many therapies that target currently plasticity. But that's not to say people aren't sort of thinking about it. People are thinking about two types of therapies: state gating therapies that hold cells in a certain state and don't allow a transition to another state potentially, and state changing therapies that actually push back from a certain malignant state back to a more benign state where the cells could be killed and are sensitive to therapy. You heard a lot about mutations in phylogeny yesterday, and this is still a question that I'm learning about. My training is also in computer science and a certain amount of math, and I'm learning as I go as well. But from what I can see, mutations can result in altered plasticity landscapes, and many mutations can sort of create the same change. So there's a convergence in terms of phenotype. So when you're looking at things mutationally, there's thousands upon thousands of positions that can be mutated. They can lead to a predisposition of certain kinds of states. So that's as far as I'm able to connect it. The good news is that this state space, with the current technologies we have, where we can measure single cell RNA-seq, single cell proteomics, epigenomics to some extent, they do seem mappable. So we wrote this sort of review a while ago, which has become semi-popular, that we can map phenotypic plasticity and the cancer cell landscape using the computational term manifold learning, which is just kind of learning the state space. What is the data that allows us to do this? Mainly, I would say it's the single cell data that gives us small variations between different cells. So the most canonical types of single cell data you can kind of think of as a large matrix, where the observations are cells and the features on these observations, they can be genes if it's single cell RNA-seq, which is maybe the most popular. I've also worked with a lot of data sets where these are proteins, for example, with CyTOF or MIBI or some of these technologies. So what you get is this large matrix, but you may have, as you've maybe heard before, this matrix is going to be somewhat sparse. All the gene counts are undercounted. There's a lot of noise. So I won't talk a lot about it today, but underlying state space mapping techniques have to be immune to noise. And actually, I will talk about it a little bit when I talk about PHATE. These days, short time courses are sometimes available, but they're just discretely many time points. So it's like 1, 2, 3, 4, 5. And a lot of what I'll be talking about is how can we infer continuous trajectories of state change. And there could be an event that's happening right here where you haven't really measured because you skipped over from day 0 to day 1, and there could be something that's targetable there. That kind of thing. So inferring continuous trajectories. This data set you'll see again, it's from my breast cancer study. And it seems like the state space, as I was saying, is mappable, and we generally see sort of trajectories or at least progressions when we map the state space. And this is from a recent colorectal cancer study. We were fortunate enough to just get a CR UK Cancer Grand Challenge grant, where the purpose of the grant is to gate the cells, like keep them from advancing to this sort of fetal state that you see here. So in order to do this, so that's kind of the problem setup of what we're trying to do. We're trying to understand the different states that cancer cells exist in, and we're potentially looking at how those transitions are made so that certain genes or markers can be, signaling markers can be inhibited to gate this kind of change. And so we've developed, I would say, in the past 5 to 6 years, so the first paper I'm talking about here is maybe circa 2020. But I mean, the first concept is maybe circa 2020. First is to map the cells into the right space, which is kind of like a precursor where you can see the transitions. And then I'll talk about how you can use neural ODEs that are regularized at a baseline to perform dynamic optimal transport to understand maybe the continuous cellular trajectories that result, how you can leverage these continuous time trajectories you inferred to learn underlying regulatory networks, and finally I'll kind of end with this idea of phenoscaping, which I'll explain in a little bit, probably in part 2. The first step that we do that we find very useful is to put the data in what we call a geometry preserving space, where you can actually see the state space intuitively and assess what the transitions could be. And here I'll be going back to our PHATE visualization technique, which was published in Nature Biotech in 2019, but we continue to find it useful. We've made many variations of it that I'll refer to sort of at the end. The idea of PHATE is that it is a metric preserving dimensionality reduction algorithm, so it preserves some kind of distance in its embedding, but it's preserving sort of a more data intrinsic distance rather than Euclidean distance. Our goal when developing PHATE was actually to create a visualization or something that could go down to quite low dimensions that preserves data geometry. Subsequently, it's been useful because a lot of generative models, like your flow and diffusion models, also operate well in these lower dimensional settings. Some of the considerations we had was that this data is hugely noisy. We don't want the noise to cloud the shape of the data. We want to have some kind of geometry in our latent space so that the distances have some kind of meaning. We want it to be able to compress it into low dimensions. And we want to allow scalability to large modern data sets. So here is sort of a toy data set. Some of you may have seen this example before. This is sort of a branching trajectory-like structure. And if you sample data along this structure and add some noise, and we didn't add that much noise, you can see a little bit of noise, and you look at how different dimensionality reduction methods preserve this data, you see that some of them are emphasizing local neighborhoods so they could shatter the global space. Then it makes these transitions and trajectories very, very hard to map. And especially if you don't have, if the distances aren't offering you something meaningful and you're getting global scrambling. There are some other techniques that can preserve some of these trajectories, but they preserve them in quite high dimensions. Diffusion maps on average preserve one trajectory per dimension. So if you had 10 trajectories in your data, you'd need something like 10 or 11 dimensions. PCA does sort of a linear projection, leaves off a lot of variability, so you could see fewer branches than there are, and so on. So this was our motivation for developing PHATE. PHATE actually has a close relationship with diffusion maps, but it is different than diffusion maps, so I'll explain that in a second. So in PHATE, again, let's suppose you have this cell by gene data. We go from the cell by gene data to a cell by cell Euclidean distance matrix. And I should say these measurements are usually library sized normalized, so divided by the total number, so that you don't get variability based on how many transcripts were sampled per cell. We change that distance matrix over into an affinity matrix using a kernel we designed called the alpha decay kernel. But I guess it has some connections to sort of like a Gaussian kernel or a negative exponential kernel like that. And what this kernel does, this illustration is showing the viewpoint of a particular cell. This particular cell has its near neighbors and its far neighbors, but then when you do the affinity matrix, you localize the neighborhood. Then you can Markov normalize this matrix, so it becomes rho stochastic. This becomes a Markovian matrix, and you can advance a Markov random walk from this. When you do that, you reestablish connections again, but this time through the data, because you're taking random walks through the data. And then at this point, each row of this powered Markov matrix has the t-step transition probability from every cell to every other cell. So every cell is sort of contextualized. We log scale these probabilities and take a symmetric divergence. And this is sort of an informational distance. And this distance, we just preserve it with metric MDS as best as we can. So why are these steps important? The diffusion step is important because this step is the most denoising of the steps. It's trying to reestablish connectivity through highly dense probable paths in the data rather than shortcut paths due to outliers. Then we have global contextualization. Every cell is represented by transition probabilities to other cells. And then we're preserving this distance using metric MDS. We showed that if you look at the original diffusion map, which are eigenvectors of that same Markovian random walk operator, then you get this kind of inner products between these diffusion components. So what we actually see is that our distance, key features of our potential distance is that it's a triplet distance. So if you're computing the distance between 2 points x and y, it's summing over those diffusion component inner products to all other points z. And the damping factor, this log damping factor that you have, increases the importance of faraway stuff so that you don't get this nearest neighbor type graph where you're overemphasizing near neighbors and just kind of pushing or repelling everything else away. And it accounts for covariance arising from random walks starting at those data points. So we use this initially for stem cell development data, and you can see.
Sorry, Smita, could you go back a little bit? I don't know if I'm the only one that is a little bit lost here. But,
Sure.
Sorry?
I'm sure not.
Okay, so I think a lot of us are lost. So could you go back even to the slide where you have the 6, A through F? Yeah. So could you maybe slow down a little bit and tell us what are you doing there? Because honestly, I sort of understood locally, but I completely lost globally myself. So
here I'm just telling you the steps of a method called PHATE that we use to dimensionality reduce and re-embed the data.
Okay, but what is, maybe you could tell us why you do A to B, why you do B to C, what exactly do you gain? I mean, if I look at those pictures, what do I learn that I didn't know just looking at A? Again, if I'm the only one, I don't need to.
Oh, I support this question.
Okay, all right.
Yeah, so I think I tried to tell you this. The first step is if you're just measuring Euclidean distances between data points, you'll become confused by outliers. So if you imagine you have a Swiss roll or something like that, you could develop a distance that's unrealistic for, so I can kind of show you. How do I get the annotations? I actually don't know if annotations are enabled. So basically the idea is if you have a cell here and a cell here, and you're measuring a Euclidean distance between them, that's a nonsensical distance because cells can't transition like that.
So is that like saying if this was on a manifold, I want to do a Riemannian metric on a manifold?
That's right, exactly.
Okay, so that's what you do. Okay.
Yeah.
All right.
Yeah, that's why I called it manifold learning, but I've had pushback from people calling it.
Well, they're not a manifold in that case, but I can see the point.
Yeah, it's data sort of sampled from a high-dimensional manifold, and I'm trying to exactly develop a Riemannian metric on the manifold. You're right.
And the diffusion thing, and then, okay, we could go.
The diffusion is approximating the Riemannian manifold by taking small steps rather than large steps. And you're taking small steps based on the local Euclidean distances.
And the color code, for instance, in C, you have this high.
I'm showing the viewpoint of a particular cell, I said. So I'm showing the viewpoint of this cell in red. And this is how the affinities come out for that particular point.
Affinities are like non-symmetric distances or something like that? Is that?
Affinities are distances passed through a symmetric kernel.
Okay, symmetrical. Okay.
Yes. So it's like a Gaussian kernel, but it has a higher power. Yeah, so these are distances that are based on random walks through the data. So exactly, they would be like manifold distances if you had an idealized manifold. Any other questions?
Any other questions? I think.
So, so I don't know if this is a fair question or, I mean, you said you were going to end up with these low-dimensional images, right? Your picture ends up with something that's embedded in 2 dimensions. But in principle, you could end up with a high-dimensional object, right? That isn't going to get a nice, if you try to preserve any kind of metric, you won't end up with a nice 2-dimensional image, right?
Yeah, well, so the difference here is there are some dimensionality reduction methods where you only take eigencomponent 1 and 2, and you're forced to discard the rest. So we're not doing that. We're solving an optimization to put all of them into whatever dimensions you say, 2 or 3. We are not the only ones that do that. The famous dimensionality reduction methods of UMAP and TSNE do this too. But they are not trying to preserve any metric when they do that. They are just trying to preserve near neighbors when they do that. So we have an optimization called metric MDS that will squeeze distance preservation into 2 or 3 dimensions. So you get the best possible estimate of your manifold of these distances preserved in 2 to 3 dimensions.
But wait, Smita, there's theorems that tell you that if you go from dimension K to dimension L, there's a minimum distortion in distances, and that distortion can be huge. So how can you?
So one thing that I will say is that the intrinsic manifold dimensionality of most single cell data is pretty low. In ambient dimensions, you have about 20,000 genes, but the intrinsic dimensionality, even if you did a scree plot with even if you did sort of a scree plot with PCA, is about 20. So you have a lot higher chance of being able to preserve these manifold intrinsic distances than you do the Euclidean distances.
But I mean, even going from 3 to 2, you're going to get a lot of distortion. So
you'll get some distortion, of course.
Yeah.
But we are trying to preserve as much of this intrinsic kind of through the data transition information as possible.
So is there an explicit criterion that says, I want to minimize the average distance difference between the Riemannian distance and the distance on the...
Yeah, that is the optimization. The metric MDS is trying to optimally preserve the distance.
Which you didn't show us the criterion.
The MDS is sort of like a package. So I'm a machine learning person. I showed you the parts I work on. MDS is an old technique that's been figured out a while ago.
Okay, which minimizes.
That's called multi-dimensional scaling. There's a classic version, there's a metric version. I just use it as a solver.
Smita, can I ask one
Yeah.
other question? Hi, it's Sam, just so you know it's familiar. So MDS typically doesn't just do 2, right? Or am I wrong about that? Like you can...
Metric MDS can do 2. That's a nonlinear optimization technique. So it has an actual optimizer. It's either SMAC off or SGD.
Sure. I didn't mean it can't. I just mean you could...
You could.
You
Yeah,
could,
and
in
in fact,
theory,
we
try
do.
to do this in higher dimensions and then just visualize in lower, right? Did you guys
Exactly.
ever
We do
experiment
this, in
with that?
fact. Yes. Yeah, I'll get to that. This
Okay.
is just the first step.
Okay.
I'm
So-
actually going to show how we do trajectories in this space. I think Michael had a question. Michael? I saw his hand raised, but he disappeared.
There was someone on Zoom who had their
Oh,
hand
I
raised,
see.
but I don't.
Yeah, no, he's one of my collaborators, Michael, so I'll catch him later. Okay, so the end result of this is that we have been able to see transitional trajectories and these other kind of geometric structures in our data when we use PHATE in a way we can't with the other methods. And PHATE is semi-complicated, but I'm happy to offline explain why the steps are there the way they are. The basic idea is to figure out basically random walk probabilities from data to data through the data. So here is a comparison on this same data set, which is human embryonic stem cells differentiating into different lineages. We know what some of these lineages are, and so we can figure out, for example, ectoderm and endoderm in a way that you can't with others. And here's sort of a systematic comparison. If you were preserving just Euclidean distances in ambient space between these cells, this is what you would get, and it kind of looks like a blob. So Euclidean distances aren't terribly illuminating. Something like isomap, which also provides distances through the data, is better. That's also a manifold learning sort of technique. But it looks at shortest paths between points, and so it's very noisy. So any outlier will cause it to kind of distort things. I mentioned TSNE and UMAP are not trying to preserve manifold distances really, and PHATE has this effect where it does preserve these same kind of distances through the data, but it does it in a cleaner way because it uses these diffusion path probabilities rather than shortest paths like isomap does. And isomap, by the way, also uses MDS at the end, so it can get out more trajectories into lower spaces than diffusion maps do. So that's a little bit about how PHATE works. Sometimes people think that PHATE is only useful for trajectory style data, so I always show them this, that it can be useful for cluster data too, but it can help illuminate some kinds of trajectories inside the clusters. So that's kind of the space we use to learn our trajectories. After that, what we're doing is what my talk advertised, which is these neural ODE models that are run on top of that learn or infer trajectories of cells. So the problem you have in single cell data is you could have a short time course like I illustrated before. In fact, this human embryonic stem cell data I showed has 5 different time points, but you are never able to follow a single cell. Those cells all die in the machine, and then you have a whole new set of cells. You don't actually have the connections between cells. And sometimes when we're not able to measure cells very frequently, like for example, tumors growing in mice, you can develop pretty large gaps between the cells. So our input data could indeed have gaps. And what you would want your neural ODE to do is sort of connect the dots, so to speak. So if you had a cell from an initial time point, you might wonder, where will this cell go to? And in cancer, this would be like asking the question, what cells from my initial tumor actually create a particular metastasis versus another? So it would be great to connect these dots, and it would also be interesting to see when these, as a population, when these trajectories are splitting and what sort of causes the split to go from A to B. This is analyzing the decision. So this is where we use our neural ODE. Our neural ODE is penalized at a population level to take a population you have at time point 1 or pseudo time point 1 and match it to population time point 2, but in a way so that the intermediate path is as smooth as possible. So I'll show you a little bit of what happens when that's not smooth. And as you do this population level matching, you're actually drawing these single cell trajectories in the wake of that transport. So our latest network
Can I ask a question? I mean, just based on something you just said, go back one step.
Mm-hmm.
I mean, you said you sometimes use pseudo time 1, but in the case of splitting trajectories, doesn't that confuse the 2 different trajectories? If I look at green and dark green at one time and light and connect that with dark green.
I don't have to split. I'm not splitting them by lineage. I'm just saying these are the cells at time 0.
No, I understand, but I'm just saying. But then you have to use real time. You can't use pseudo time for a splitting trajectory. That's a question. It wasn't a statement.
No, I can use pseudo time.
People think about pseudotime combined with actual topology, sort of. They don't, when they say pseudotime, they don't necessarily just mean the actual distance along a trajectory.
No, they mean the distance.
terminology.
They mean the distance between. They identify as the same time 2 different cells that look the same from the point of view of the gene expression.
It
Roughly
means
speaking.
so. It really means distance along some kind of process, right? Like you're saying, like so that
Right.
they're similar. But I'm just saying that when there's an actual split, like you're talking
Right.
about, right? We actually kind of abuse the term pseudotime because we're sort of thinking
That's
about
sort of what
that
I'm
as
asking,
distinguishing along
Because I've never
branches.
understood how you use
Yeah, it's a
pseudotime when you have
complete
splitting
abuse of
trajectories.
terminology.
Okay.
Totally
Okay.
have to think about it as if that's implicitly we're also thinking about the branching structure
Okay.
or whatever it is there. So that's just like a translational kind of
Probably, but I don't know the other meaning of it. So yeah, there is some art that's associated with progression. Like you could have enrichment of cancer cells somewhere, or here sometimes I have real time point. So a real time point. So even with crude time points, we can.
I guess
come
I
up with-
had a separate question, which is, my impression, maybe I'm getting the wrong impression, is you're doing the optimal transport on the reduced 2-dimensional data as opposed to the data in the originals.
I'm doing it on a reduced 5 to 10 dimensional data.
5 to 10,
because the
okay, so
original
not 2.
space is.
Okay, it's too
the
big, but
original space-
people
yeah,
do
it's too
10,
big.
20, or whatever it is, so that's what
Yes,
you're doing also.
exactly. Yes,
Okay.
and that's why I said I use the 10, 20 dimension. I don't just use it in 2 dimensions as
Okay,
somebody.
that's,
corrected.
that was the question. Okay, thank you.
Yeah, so, and in fact, I draw my neural network as if it's getting like a 10-dimensional input or something like that, just to clarify. So we use a PHATE regularized autoencoder, and the reason that's good is it solves certain problems. It puts you in this space where it's denoised and you can clearly see the trajectories, but at the same time, something you can't do with methods like PHATE, isomap, diffusion maps, or UMAP, decode them back into the original space. So I'm using a PHATE regularized encoder-decoder framework here, and the regularization is to also preserve PHATE distances. And sometimes that's better done if you train this in 2 parts. The encoder is trained to preserve PHATE distances on the training data, and the decoder decodes from there. So once you put your data in that space, we learn a neural network. That neural network learns a time parameterized or a time instant derivative of the cell state. Recently, and this is not in our published version yet, we also learn a time instantaneous growth rate. And so that means that the cells are allowed to proliferate or die from any state.
How do you get those? Can I ask? I've thought about this. Like, how do you, because they tried to do this a little bit with previous OT work, and it seemed like it was really hard to estimate those.
The neural network learns it.
So it's a parameter that you fit along with everything else. Okay, got it.
Are you going to show us
That's the whole thing the neural network gives you. How is it penalized? It's penalized at the population level. At the population level, it sort of has to match the next time point. So that's where that it's performing the optimal transport. And so the regularization to preserve PHATE distances just looks like here's the difference between the high dimensional potential distances versus the embedded distance. And then the ODE solver, that's what the reason this is called an ODE is because this gives the derivative, and you integrate the derivative over from time point 0 to the next time point 1, and you can just even check with KL divergence or penalize with KL divergence how well this matches. But one thing that should be noted is there's a difference between the static optimal transport or static KL divergence I would be using to match this versus the dynamic optimal transport that I'm trying to get the paths to satisfy. The static optimal transport problem, as you guys know, it's sort of a linear programming problem. If these two were one-dimensional distributions, you would get a joint distribution that's a transport plan, and you would be penalizing the transport plan based on ground distance. So if we were trying for the whole solution just to be a static optimal transport plan, then you have no control over the intermediate path, which is exactly what we're trying to get. So these might, result in, circuitous paths that we don't have a lot of evidence for that have high frequency fluctuations. Instead, we are adding a simple L2 penalty on the magnitude of the derivative that comes out from here. This once you do that, you get these much smoother paths that there is that's not implying some kind of walk through empty space, for example. And in our manuscript, we do show that once we do this at convergence, if the magnitude of the derivative is indeed small, then we match the definition of what's called dynamic optimal transport, which was developed in 2000 by Brenier and Benamo, and we have this geodesic manifold ground distance. We do add some additional losses to this base optimal transport penalized ODE. For example, this would be an implausible path. And we want to penalize against it. So we use a density penalty. We penalize the distance to the nearest neighbor so that as much as possible, it tries to take a path that goes through the data. And as I mentioned before, we have a growth proliferation network so that we don't force matches between cell types that are not connected. We will just allow cell types to die or proliferate. And this also gives us some interesting information. At the end of the day, the optimal transport sort of looks like this. And you see, as I said, you're kind of pushing the population along and you're getting the single cell trajectories in their wake. What can you do with these single cell trajectories? One interesting thing to do is go to the end states. You can see these end states are different. And then ask, how did they get here? What happened along the way that was different in some of these states? So you can see certain markers go up. These are gene expression values. Certain gene expression values go down. And there's a whole variety of behaviors that are happening that are different for the different lineages.
How do you know those are Smita, how do you know those are end states rather than beginning states?
We have time here. So these are human embryonic stem cells. Always all our cells in our body start as embryonic stem cells.
Right. Yeah, so in other words, you do it in advance for biological reasons, right? So in other words, you sort of know a bit about the structure of the trajectory to begin with. You're trying to infer the rest of it.
Yeah, I had 5 gross time points that I'd measured. This was the time point 0.
Oh, okay.
Okay, and then that constitutes this. And if you don't have that, you can run a wonderful software from Fabian Theis to give you an arc like this. So there is some prior information about the general direction of the trajectory, but not about the details.
So along those lines, how do you compute a derivative?
The neural network computes it.
Oh, I mean, on what basis does the neural network compute it? What is the idea?
On the basis of how I penalize it.
Penalize what?
The neural network. I train the neural network based on a penalty, and it figures it out. And that penalty is that optimal transport matching penalty. So it computes the derivative that will cause the cells to match with the next time point if I integrate them out to the next time point.
And you use automatic differentiation to compute the derivative.
What's that?
You use automatic differentiation to compute the derivative.
So the matching has to do with the distribution at time point 1 gets mapped to a distribution at time point 2.
That's right.
And you're actually mapping individual cells at time point 1 to individual cells at time point 2.
No, I'm doing population level transport. So I'm penalizing with static optimal transport.
You don't know the individual cells, so you can't do that. All you have is stochastic information.
You don't have the cells all died from time point 1 to time point 2, so we don't have the matching.
You have distributional information, so you're matching distribution to distribution.
So what are we computing the derivative of?
Cell states.
Cell states. Okay.
So there's
So the neural network takes in a cell at a particular state, and it'll tell you how it's changing at future points in time.
But it's being penalized based on population data,
Exactly.
So is
That's
it
right.
not based on,
Yeah, and is it so you sort of have some kind of implicit representation of the system, right, in the ODE,
Mm-hmm.
and so you could think about computing differentiation, through this implicit representation of the ODE system, right? So it's like it's not.
So something like expression levels are proxies for cell states. And then
Yes.
computing
So
derivatives
cell
of
states are defined, as I said initially, based on this kind of data of gene expression or epigenome. Yeah. So it's a low-dimensional representation of the genome or the epigenome. So one thing that we've been doing, and I'm about to end part 1 now, is we have so many genes. So now that we have these cellular trajectories of any particular cell, we can decode it back out to the gene state. And one thing we've been doing is ordering these cell genes by when they peak along the trajectory. One of the reasons we're doing that is so that we can see which genes come up first, which come up next, which come up last in any type of a transitional process. And the one example I want to give you is this triple negative breast cancer data, where we have about 5 time points of information. These are breast cancer cells that are initially seeded like this, but some of them grow into tumor-like clumps. This is what the PHATE looks like. And one of the things we do is we run this MIOflow methodology to understand specifically 2 different types of trajectories. We have a lot of other trajectories, but our collaborators are particularly interested in this orange versus green trajectory. So from the initial cell culture, some cells just die, and a subset of another group of cells, even though they start pretty similarly, one goes to this clumpy tumor formation and one goes to cell death. And you can tell this by the mitochondrial score. And we'd like to understand why.
Sorry, Smita, we're sort of running into the coffee break. So would this be a place that you could just pause and then in 10 minutes we'll resume?
For example.
Is that okay?
Because
it was supposed to end at 9:30.
Yeah.
Okay. All right, so thank you so much. Clap, clap, clap. And,